Susan, you may carry on with the training.
A very good morning. Welcome once again for our today's webinar. Today we are having day 3. I'm going to continue from where we stopped, and I complete my part, and then Professor Susan will resume from where I would have stopped. I presume that roughly I will use the first 30 minutes, and then she will take over. Participant, I would like to ask, can you please type in the chat if you try to attempt the homework and you successfully imported the data? For those of you who have logged in, if you attempted the exercise and you successfully imported the data. Okay, great. Quite a good number. So I'm going to proceed on to what we are supposed to have today. But we are going to keep using our diamond data that we imported yesterday, or in case somebody used a different name of the data set, lab 2 data set, for those who use the read_excel command. Okay, let's proceed from where we stopped. I'm just sharing my PowerPoint, and then we continue. Looking at the recap we had yesterday, it's good enough that most of you managed to import the data. We looked at data manipulation, and we looked at the different things that we can do under data manipulation, the subset, how you can rename a variable, how you can select some variables in case you do not want them. And then lastly, we wanted to move. We did some descriptive. We did some descriptives yesterday, but the little ones, but today we are going to proceed, and then I only do the graphical bit. We still learned on how to use as a factor in case you have a variable within your data that is categorical in nature. So you have to alert R and tell it that, you know what, the variable that I'm going to use is categorical in nature, so use the as a factor variable. I also have to keep you notified that we always use a $sign in case you want to attach a variable on a data set. That one, it's a command within R, which is in view that we need to use throughout. So we looked at this part yesterday on how to get the class. So today, let's look at what we call univariate analysis. Just since we are going to move into the graphical bit, then the bivariate will come since we still have more time in the week. When we talk about univariate analysis as our the data manipulation number 6. With the univariate, the way you hear the word un1, sometimes you can consider one categorical value. And when you consider one categorical variable, for example, we had quality or color, most of the time, the method of analysis that we use is a frequency. You run frequency tables and get percentages. And when you want to visualize, you can use a pie chart, or you can use a bar graph. And in case it's a scale variable, in our data, all the variables that were numerical, either integer and so on, all those are scale variables. And in case you want to run a univariate analysis on them, you run a summary statistics. Yesterday we looked at a summary command, how you can use a summary command to give you the results. And when you want to do the method of visualization that we use for a univariate, you can use a histogram. This other bit of the bivariate, we'll look at it as we journey through the week, because it also has a separate part on the regression that we'll look at it. But maybe just to echo it, in case it's a bivariate, it implies that you're going to use 2 variables. But still, like I mentioned earlier, you need to understand the nature of your data set. In case you're considering a one categorical variable and one scale, most of the time, the right analysis to use, the method of analysis, will be an ANOVA, or you can call it comparison of means. And graphically, you can use a bar chart or a line graph. And then in case you're considering 2 categorical variables, for example, in our data set, we had quality and color. If one is a dependent and the other one is an independent, most of the time we run a chi-square test. and graphically you can use the bar chart, which are either clustered or they're stacked together. And in case you're having two continuous variables or two scale variables, you do a correlation coefficient, and graphically you can run a scatter plot. So that particular part we'll see more in details as we move on. So yesterday we looked at the simple descriptor analysis and how you can use a table command. I remember the table command when we are running it on the categoricals, they gave us the frequencies and then the summary for the scale variables. All these things for a case of the descriptor analysis, most of the time what we are doing is that we do the measure of central tendency and then the dispersion, but we looked at that yesterday. So now today, I'm going to introduce to you now how do we do some graphics within R and then to support our analysis such that it looks very well. Most of the required packages in data visualization, we've already installed them earlier. You can use the Tidyverse, you can use the ggplot2, and then the one plus other packages that we had. Remember, we are building on, depending on the package that you want, it's what you install. So now I'm going to move to my R Studio and then see how do we get this process, because they will make more sense here. They were just drawings. So I'm going to close this and then move to R Studio. Okay, now let's move to our R Studio. Let me clear all this. If you want to clear your console, you type Ctrl+L. Then it becomes you get a clear window. And if you want to clear your environment, what you do, I shared it yesterday, you type remove, and then inside you put list equals to ls. We looked at this command already. And then what you do, in this case, you can enter it, or you can also write it in your R Studio such that you're able to store it. When you look at what I've done, you see that now my environment is empty. So I'm going to start doing everything afresh. Okay. Remember, the moment you want to do that analysis, all you need to do is to import your data. I'm using the data that we had earlier, the one that we used yesterday. What I'm going to do, I'm just going to import my data again, and then after that, I'll continue with the analysis. And I kindly ask you that you also do the same. Let's import our diamond data again, such that we proceed. Okay, remember, this is the same thing that we did yesterday. What I'm doing right now, it is just to rerun. I need a kind reminder, please rerun your libraries. Just go back in the same R script and then run the libraries that we installed. Okay, then after that, run all the libraries and ensure that they have successfully loaded. If you don't see anything in your console which is red, then that means that automatically that your packages have been loaded successfully. So now after that, I want to import my data set. I'm using the same script that we had yesterday, but I'm just building on. So don't get confused, it's the same thing. Yes, now I imported my data set, which is diamond. For those of you who have your data set as lab_data2, please maintain that particular name. It doesn't change anything. Most of the time, it's always good to remind yourself on the structure of the data set that you're working on. So when you run the structure, it will tell you how your diamond looks like. In this case, it will show you that your data frame has got 100 variables and has got 100 observations and 7 variables. with the variables that you have weight, quality, color, price, length, width, and then depth. When you look at their class, it shows you that weight and price, length, width, and depth, they are numerical in nature, but quality and color are character. So you need to specify to R that, you know what, quality and color, they are factors before you proceed with the analysis. Because when you miss it out, you have to make sure that you do it at a later part. So we will do that as we move on. So remember what we are doing today. We want to see some graphics in R. I can automatically tell R that that quality and color are categorical in nature, then I proceed, or I can do it at a later part. In case I want to do that, I do the same thing that I did yesterday, and then you use the command as a factor, but you make sure that you attach in the data set. So in case I want to run a remember from the previous slide, we said if you want to run a histogram, then it implies that you can do it on a scale variable or an integer variable or a numerical variable. So what I did, I said that I want to run, I want to have a histogram, because I know my weight is a scale, or it's continuous in nature. So I can run a histogram because that is the mode of visualization that I need to use. So I use a command which is hist, okay? And then within the command, I specify the variable which I want to plot. So what I do, I tell R that go to the diamond data set and then pick a variable weight and give me the histogram. For example, in this case, you put the name of your data set. I put my name as diamond. How do I call a variable in a data set? I always use a $sign. To kindly keep that at the back of your mind. We always use a $sign, and then you put the variable that you have. In this case, I can pick weight, I can pick length, I can pick width. So after that, after putting my command, in case you want to see more details, remember we said we can use help, and then R can support you on how to use a certain command. So when I reach here, I can click run. And when I run, I guess when you look at your right-hand side, the bottom workspace, you're seeing a histogram. But in this particular case, it might not look so clear. So what I do, I zoom. The moment I zoom, I'm able to see how my histogram looks like. I got hist, then in bracket, I told R from my diamond data set, draw for me the histogram for weight. Okay, that's what I did. You can do the same for price, because also price is continuous. All I need to do, still I use the same command, that hist in bracket, go to my diamond data set, pick price, okay? And then later, I just run it in the R Studio and I get the result under the plot. For you to view it properly, where you are, you can say zoom, and then you see as you're plotting, it gives you the frequency and then the price, depending on the price, and then how much has been paid. All right, now that is for the case of numerical variables. What about for the case of categorical ones? What do? We said you can either use a pie chart or a bar graph. But remember, I need to tell R that quality and color, they are categorical variables in case I want to have a pie chart or a bar graph. So I need to attach them to R. I will say diamond. That is my data set. I'm commanding R that my quality, the quality variable, it is a factor. So I need to tell R then diamond quality. Okay, I'm going to do the same. For color, then assign it as factor. Then in brackets, I put diamond and color. We saw this yesterday. So the point is, the moment I use as a factor, remember I said diamond quality. I started with this because I want it to be performed in the same data set. That within my diamond data set, the variable quality, consider it as a factor or categorical. Also, within my same data set, the variable color, consider it as a categorical. So what I do, I run everything in the R Studio, and everything is executed into the R console. For me to confirm that it has been done rightly, I go back and type the structure and check whether now things have changed or they are still the same. So the moment I type structure and then I run, I check in my console. Now it will tell me when I come to quality that right now it has, instead of character, it is now being read as a factor. And when you come to color, it's no longer character, it's now being read as a factor. So now that I've commanded R, then I now, let me move on to do the visualization. So remember what we are doing, that in case you're doing a descriptive analysis on a categorical variable, I can run a table, okay? If you remember yesterday, we used the table command. You say table, and then you write diamond. I call my variable quality. So I can write that, and then I run. The moment I run and I look in my R console, I'm seeing that I'm having the attributes of quality. What is fair, good, ideal, premium, and very good, plus their respective frequencies. That's the fair is 5, good is 8, ideal I've got 39 diamonds, premium they're 35, and very good they're 13. However, sometimes I can create an object to say that, okay, I can create an object called count. Remember, an object is a naming, so you can call it any. And I assign it to be equal to the whole of this, because I might need to reuse again my object, like the way we assigned on the full data set. So I can assign on my table, and I assign any name. This time around, I've called it counts, but you can call it anything. It's the same. I'm saying that let counts, my object, or the name that I've proposed, be equal to this particular expression, table into diamond. I want to get the frequencies because it looks a very long line, so I want to condense it. When I run this line, I look at R. Okay. Okay, some participants are saying that I'm fast. I'm going to slow down. Let me ask in the chat, have we gotten how we can draw a histogram? Okay. Okay, let me go back such that we move again on the same pace. It's all right. Today we are looking at data visualization, okay? Sometimes some people will say that graphics or a diagram speaks far much better than the wordings or than the figures. And it's always good, for example, in case we are writing out our projects or you're writing out a manuscript, and then you can also present some of your results using diagrams or graphics. So I'm using the diamond data set we had yesterday. Let me ask in the chat, have you successfully loaded your, imported your data diamond into R today? Okay. Make sure you load the libraries again and then import. All you need to do, just go back in the R script you used yesterday and then load the libraries and click run. Then after, call your data set in R. If you already have your data set, and depending on the variable name, the object name that you gave it, then we can move on. So I'm asking again, what we did yesterday, load your library. When you load your libraries, if using the same script we had yesterday, what I did, I've not changed anything. I don't need to install again. I went and I loaded my libraries because remember I told you that is the assignment we had on Monday. I can highlight everything instead of running line per line. I can run line per line, it is okay. Or I can highlight all my libraries and load the packages for today. And then I run. That is the part. Since I've loaded my packages, then I'm good to go. I'm good to go and now load my data. The next step is I need to import my data from Excel and then I import it to R. Okay, please load the packages and then import the data and we proceed. Yes. I'm using the same script we used yesterday. Please don't get confused, the module1.r, which you already have. But you can also write in a different R script on your computer such that you're able to follow closely. Okay. Remember importing our data, you can either use the read_Excel or option 1 or option 2, you can go to session, then you choose the working directory by when you're going to use the read.csv command. Using the same approach we had yesterday, when I use the read.csv, when I've already set my working directory, I'm able to import my data in R. So I'm going to run. I'm going to ask in the chat, have you successfully loaded your packages and imported data in R? Okay. Great. Please import your data in R. Still to remind you, we shall have support sessions after here. So now let's move on. Depending on the object that you've given your data set. If my object name that I've given my data set, I want to call my data set diamond. If you imported the one in Excel, it automatically generates for you the object, depending on the name that you gave your Excel sheet. Okay, now what I want us to do is now to move on how we can have some graphics from our data, because all this is important to get familiar with the data that we are going to use throughout. For my sessions, we'll be using this data set till we get to the final analysis on the last stage in our learning. So, and then I said, in case, when you look at the variables you have in your data set, it's always important to understand the different level of scales. I saw that within the data set, I've got variables that are numerical in nature and others are categorical in nature. And this one gives their picture on which kind of visualization that you're going to use. So we are going to start with the continuous variables. And when you want to present the continuous variables, you can draw a histogram. And I'm going to go in the chat and ask, can I continue? How do you want? Which chat? Okay, I'll show that. All right, okay, let's proceed. Now, we want to draw a histogram. Since it is continuous, we are going to use the command hist. You can check the structure of your data set. The structure of your data set, it is the one that will support you to know the different classes or the type of the variables that you're having. In case of the variable, the class they are giving you, for example, if you look down here in my console, if you're seeing it, the class I'm telling me weight is numerical, character and color, quality and color are characters, then integer numerical. So the continuous ones, we use the histogram. And now, if you want to get more help in R, we said you use the help command, and then it will give you more details on how you can use that particular command. You can say help, in bracket, you put what? You put hist, and then you run. Even before you run, it tells you that this command computes a histogram of the given data values. Okay, so it is always there to support you that when you want this, this is what it's going to do. Right, so using the command hist, I go that from my data set, which is diamond, depending on the name that you gave it, I want to draw a histogram for weight. You can draw a histogram for length, for width, for price, because all these are continuous variables. Okay, let's take a look. In case I run that, in case I click at the end of the line and I run, the output is being seen if you look at your right-hand side by the bottom workspace. This is where you see the figure. But for you to see it clearly, why my figure is not seen clearly, because I've analyzed the different boxes. You click on zoom, okay? And then the picture comes out very well and you're able to see what you're having, that you're having a histogram for the weight, okay? And you can be able, I hope you're all seeing this, where there is an export icon. In case you want to save your figure, you come and click on the drop box. They give you different alternatives. Do you want to save it as an image? Do you want to save it as a PDF? Or you want to copy it to the clipboard? So all you do, you pick any option that you want. In case I want to save it as an image, then it will ask me, where exactly do you want it to be? It is here, it is giving you a preview of how it looks like. And then the image format, it's telling you, do you want it as a PNG, as a JPEG, anything, or an EPS, okay? In case I leave it as a GPS, then I come to the directory like what we did yesterday. I'm commanding R that save this picture in this particular folder. But since you already have the working directory, R gives it to you already that, you know, this is the working directory that I have. Then you can say save. And the moment you click save, you go back to your folder where you're doing your work, and then check that figure will be there. I'm going to the chat to ask, have you been able to see how we can plot a histogram? Sorry?
Some people want to put their original data diamond, they can't fit it. All they can see is diamond failure, diamond what, so how can they use their original data?
What is original?
The diamond data, which is not manipulated. That's what they want.
From the R website. Okay, let's continue. Now, you can do the same for weight, you can do the same for price and so on. Okay. Now, what about in case somebody is saying that, can I repeat how to save the figure? If you want to do that, look in the box where you're seeing your plot, where you're seeing the figure that you've gotten, all right? There is, after the zoom, there is another icon which shows export, and it has got a down, an arrow pointing down. So you click on it, and then it will ask you different options that you might wish to save. Do you want to save it as an image, as a PDF, or you copy it to a clipboard, and then you do other types. Then I said I can click it as save as image, or I can save it as a PDF. Any option that you pick works. So in case I pick that, then it will ask you, where do you want to save this picture? Remember, we looked at the directory yesterday, and if already the directorate is set, it will just put it there because we did it fine, that I'm saving everything under module 1. And it's even asking you, what file name, what name do you want to give to your plot? You can, for example, say, no, the file name I want, you can say it's a histogram. And then all you need to do is to click save, and then check in your folder where you're doing, where you set the one that you set your working directory, and then that figure will be saved there. All right, now let's move on. In case we are looking at categorical values, categorical variables, remember we said you need to tell R that variable so and so, it's categorical in nature. And for you to do that, you use as .factor command, okay? And this is what I'm having here. But you want to do everything within the data set, that within my data set diamond, please make quality categorical, make color categorical. That's why I start with this expression, and I say diamond $quality, that within the data set diamond, pick my variable quality, and then read it as a factor. For you to do that, you use the as factor command. You do the same for quality, you do the same for color. Okay, we had that part yesterday. So now what we want to do is that we want to plot a bar chart for quality and color, because for them we know that they are categorical, and for you to present categorical variables graphically, you can use a bar chart or a pie chart. I'm going to go. I'm going to go in the chart and then ask, can we proceed now on how to draw a bar chart? Okay, fine. Now we are going to consider quality, okay? But remember, quality is categorical in nature. I've already told R. So for me to see a summary statistics or the frequency, I use the command table. What we did yesterday, it is table, and then in bracket, I need to put in, you want the frequencies of what? Because it's like from the frequency of table, I use the table command. After opening my bracket, I command R that from the diamond data set, I want the frequency for table by using this. So this is the line. And then after that, I can run it. The moment I run it, I'm able to see my frequencies in the R console, telling me the different frequencies for my attributes that under quality. Because remember what we said, that for a categorical variable, it has got attributes. Like you say, gender, my gender can be a variable, but my attributes are, you're either male or a female. So it's the same thing that is applying here to quality. Now, instead of for me to move on to like moving throughout my data, quoting this very big sentence, I can assign it a variable that is very simple that I can remember. And I say, okay, let me use a count to mean the whole of this particular expression. So I get an object, any name that I want, it doesn't matter. I used counts. So in case I've got counts, I select counts be equal to table, diamond, $ quality. When I click at the end, I run that. But you know what you're seeing now in the R console, it has not given me the result because I did an assignment that counts is equal to table in bracket diamond, then my $ quality. Okay, but now in case you assign an object, for you to see what is inside that object, you move to the second line and type it out only. So I went to the second line and what I did, like, okay, I want to see the inputs of quality of counts. So you type the word counts and then you run. The moment you run counts, you look in your R console and then you'll see the output. So I know that within the counts, there are frequencies for quality from my diamond data set. So now, before we proceed to plot the bar plot, are we on the same page? Okay, great. Fine. Now let's move on. We want to have a bar plot. If you want to, it's always good to use the help window because it supports you to understand what exactly that you want to do. For example, in this particular case, if I want to plot a bar plot, I can even come into my R console and I say bar plot. Sorry, I can ask help. I say help in brackets. I want help on a bar plot and then I enter. The moment I enter, I come and look in the right-hand side, the bottom workspace, and I check. They're giving me details on how it looks like. And then they tell me also the usage. For example, in this particular case, they will tell you that within the bar plot, you have to put your data. This is now this time around, they put an example, for example, of the height. And then they are putting also other arguments that you can consider in case you need them. You only pick an argument that you want. After seeing the help, you can further go and then they're asking you about main means title in case you want to label. In case you want to color it, you use call, and then they will tell you, you specify. When you say null, it means that no color. The moment you put a color, then you mention the color that you want. So it's always good to come and use the help command because it helps you to explain the different arguments or the usage of a certain command. Here they are saying that the height was either vector or a matrix of values to describe. In our case, it is going to be counts. Now I'm going back to my R Studio. Within my bar plot, I'm having counts first. That is I want to draw the bar plot for quality. Okay, I want to have a title for it, and then I want to label the x-axis. If you want to label the y-axis, you put y label. But all these arguments, you can get them from the help command and you pick what you want. And then all I do, I run this command and see what I get. Okay. Now when I run this command, I can zoom out and see how my figure looks like. Okay. This is what I have. Remember when we ran the frequencies, we saw that fair was 5. So on my y-axis, I've got the frequencies, and then on my x-axis, I've got the different kinds of quality, the quality type. I've got the fair, so you can read here it's 5. I've got good, and then you check the ideal, the premium, and then the very good. If you see that you can't see it very well, you can always maximize and then see the plot clearly. The saving part, it is still the same. you can do the saving as I showed previously. So that is where we are. If you want to check your plot, always ensure that your plot will appear under the plot window. The moment the line runs successfully, you see it under the plot window. But it's always good to ensure that you load all the packages that we have for today. If those packages are loaded right, I wouldn't expect you to have any challenge. Okay, I'm giving you a minute, and then you also plot. Can you have a bar plot for color? Participants, can you have a bar plot now for color? We did for quality. Now let's try the one for color. And then I'm going to ask in the chat if you've been able to successfully have a bar plot for color. Use the same command. All we need to do, instead of quality, this time around we put color. They show us the frequencies. You can create an object in case you want, and then use a bar plot command. Have you been able to draw the bar plot for color? Try the one of color and then respond. Okay, great. I'm seeing some positive responses. You're saying that my command is too long and you can't see it. Remember, you all have this script with you. You already have this script as a module 1.r. So this is the command that I used. All I need to do is to either put my object that I've used. Why I'm creating an object, it's because this one looks to be, this expression here looks to be too big for me to put it here. But in case you don't want to create an object, you can still put it here because before you plot it, it's automatic that you have to tell R that it's categorical. And that's how you're supposed to do it. Okay. Now, there are different ways on how you can have your bar chart. We've just looked at the first one. However, okay, let me ask in the chat. Color done. If you want to edit the title, you go back to your command and write any word that you want. You just come here and then you delete what was there, for example, and you write color. All the edits are done in the R script, and then you get the output that you want. Edits can be done not directly on the figure, but you do it in the command, and then the output will show the corrections or the changes that you've made. Okay. I'm glad that many of us have been able to have the color. So let's push on. Where is it? Okay, let's proceed. I was saying that sometimes you might have various ways that you want to see how your plot looks like, especially maybe you want it in color form, or you want it in a certain direction, whether it's horizontal, whether it's vertical, and so on. So I'm hoping that you were able to load the library AP Display. This one is just to support you in on how you can display your graphs. And I sent it on Monday that let's have that library loaded. I'm not going to load it again because I have it already. However, what is interesting with this library is that it gives you the frequency, it gives you the percentages, the cumulative, then the cumulative percentages, and then also it displays the data. I'm going to move on to this part and I explain it closely, and then I'll inquire if you've gotten it. However, if you want to do that, in this case, if you want to get everything at a go, we use a command that is tab1. Okay, tab1, it's an inbuilt command within the AP Display package. So you are able, it gives you your data in a form, it sorts your data in descending order. You can tell it that give me my data in descending order or give me my data in ascending order. And you can still inquire for help. How do I use this? So what happens? The first thing is that when you're using the tab1 command, you indicate the variable that you want. Since tab is from table, so it implies that it only works for categorical variables also. So you first display the variable that you want. So this is my variable tab. Go in my data set diamond and only consider the quality variable. The other argument that this command has, it is sort.group equals, it's an argument in this command. So you either tell R that sort my data in ascending or in descending order, whatever that you wish. So in case I indicate it as decreasing, then I close my command. Yesterday, I saw some people were having challenge that the codes were not running because you did not close the bracket. Everything has to be done within, in case you have an open bracket, you have to make sure that you close the bracket. Otherwise, you're going to keep on getting errors on something very simple and then you get frustrated. So when you have an open bracket, make sure it is closed at the end. Okay. If you need more help, you just go into R and then you say, okay, what do I want help on? For example, I can say I want help on tab 1. Even it tells you that on tab 1, it just displays multiple ways of a layout of the same tabular data. So it's about a layout. There's nothing much. In case I click at the end and I run, you see what happens? Look at my output in the R console. What it does, it gives me the frequency for my attributes. It gives me the percentages and then the cumulative percentage. So in case I'm writing down my data, for example, in a publication, because most of the time I need the frequency and then the percentage, I can quickly pick that. But it doesn't stop here. It also gives you a plot. You have to have two outputs. So in case I look at my plot, what happens? This is what it is giving me. It is showing me my data. It plots the percentages on each box, and then it renames everything in decreasing order because it's what I asked for. So it starts from 39 and then it goes down up to fair, and it also colors my my plot. Okay, so I'm going to ask again in the chat. Can you have a tab? This one was for quality. Can you try out for color and see if you're getting the output, whereby it has to give you the output, which has the frequency plus the percentages and the cumulative percentages, as well as the diagram or the graphics. I'm giving you 3 minutes to try that. Then I'll ask in the chat if you've been successfully done it. Somebody's asking if my data is different from yours. It is not tab 1. It is the tab is small t. If you're working out, use tab small t. R is case sensitive. It will tell you that it doesn't understand what you're doing. If it is not displaying, make sure that AP display package has been installed. Otherwise, it will give you an error. If you've not loaded that, if you don't have the library for AP display, you will not get the figure. Okay, done for tab color. Great. Okay, so I do one, you do the other. What about in case, remember that when you look at the plots that we are getting, they are all vertical. What if I want to get my plot when it is horizontal? What I do, if you look, consider the same command that you've used, but you just need to add another argument. You say horizontal equal to true, and then see what is going to happen. Because in case it is false, in case I put false here, it will give me the result as same as what I got previously. That's why I didn't even indicate it. But what about in case I want my figure to appear horizontally? I tell it, bring out my outcome, but I want to see it in a horizontal format as the layout that I want. In case I run that, what is going to happen? Okay, with this, I need to sometimes when it brings that error in the plot new that the margins are too big, most of the time it is being caused because I enlarged the formats. But I want somebody to try that and then tell me whether it has worked, because that is the problem, because I'm using a different font. Have you tried to, in case you've drawn it? Yes, great. Yeah. Exactly. So that is the display. That error is being caused because I enlarged my window. All right. So that is also another format that you can use. Okay. And the more you work with R, that's when you're going to see that these things become part of you. I think that is enough in case you want to have a bar plot. What about in case you want to present it as a pie chart? So in this particular case, in case you want to have a pie chart, it is always good, like, still ask R for help, that please help me on this. How does this look like? Just to take you back, I'm still going to use quality. If you look in my console, okay, if you look in my console, my ideal, when you look closely, ideal, I'm given 39. Premium, I'm told it is 35. And then very good, it is 13. Good, it's 8. And fair is 5. So now, in case I want to draw a pie chart for this, what I'm going to do, I'm just going to extract my frequencies in form of a vector. Remember when I talked about the different data types, apart from the data frame, in case you don't have a big data set, you can also represent your data as a matrix or as a vector. So what I do, I went and if you want to put figures together, you can concatenate them using this is the command, C then bracket. It's a command that you're putting things together. So now what I'm going to do, I'm going to write my frequencies for the different, the categories for quality as a vector. I've got 39, 35, 13, 8, and then 5. So this is what I've done, which I say it's 39, okay, 35, then I've got 13, I've got 8, and then 5. So this is what I'm putting them together. Now for me to make my work easy, I create a variable name because I want to get a pie chart, and I say, okay, let me give it an object called slice. That's why I created this, but there's nothing that is hard that I did. To put them together, I said, let my slices, the slice and object, be equal to what I'm having here, to my concatenated output, which in this particular form, it's like a vector. Okay, you can even run this line and check. What is happening? Sorry about that. I'm going to, I write table, then diamond, and I put quality. Remember, when I run this, I want to pick this into a vector. I concatenate this together into, I use a slice object. And that's what I'm getting. Remember, in case you use an object, for you to see the output, you have to run it. But if you don't need it, you don't want to see the output, you can leave it out. So I can run my output, and then it will tell me that within the output, I've got 39, 35, 13, 8, and then 5. So what I'm going to do, in case you want to attach labels on a vector, it is the same thing. You still concatenate. But this time, as you're concatenating, instead of figures, you use the respective words. For example, I can say C, I concatenate, and I say 39. 39 was for ideal. And then I'll start with that, and I put it there. But when you're using the wordings, you have to put them in inverted, in quotes. Okay, I do that, I put comma. So it implies that as I run 39, it's for ideal. The next is 35. My 35 is premium. So I come and write premium, and then it will become my next. Still within, then I'll paste it there. I separate with a comma. Next is 13, and 13 is very good. So I'm just copying everything, and then I paste it up there. I copy, and then I also come and I put it there. Then next is 8. My 8 is good, okay? I come and I copy good. I copy. I also put it in quotes. Then I paste it. And then lastly, in the first one, it's 5, which is quality, okay? I come and then I pick fair and I paste it here. So it is the same thing, such that as still, then these ones are 5. Also, I've got the categories. So it is what I did down here. You shouldn't get confused. That I created an object called labels, which I labeled LBLS, but they were labels that are directly linked to my slices. Now, having gotten that, for me to produce a pie chart, the command is pie. This is the command. The command is pie. And then within the data structure, what comes first is my vector, or it's the data that I want to plot. The data that I want to plot, it is this one that I concatenated, and I labeled it slices. So it's what I bring here. The next argument is I want to attach labels. So these are my labels. So labels, which is equal to LBLS. it's equal to that. I also want to put a title in bracket, quality of diamond. If you don't want to put the title, you can leave that out. And then you run. So I stopped here, then I ran my labels. And then after I ran my pi. Let me ask in the chat if you've been able to get a pi from that end. Somebody's asking, why do I arrange the values in ascending order? If you don't want them to be in ascending order, you can put, you can leave it, or you can put increasing order. Like I said, it is just a layout on how you want your picture to look like, but it is not in any way restrictive. If you don't want it to have that format, you can go with the usual one that we know. So have you been able to get all right, let's move on. Hope I'm not fast. Can we continue? Okay, the pie chart is not clear. Remember, we said you can zoom. If you zoom, if you click on zoom, it will. You will get, okay. Yeah, this is it. So we can have it is showing you the ideal, it's white, the fair, the good, the very good, and then the premium. Always use the zoom in case you want to see it clearly, and you can also save it in the different formats that you want. Okay. The second command, it was adding percentages, but it's the same thing. So on the previous plot, in case you want to add percentages, it is the only new command that I brought in, which was actually, it was slices, then over the sum. You know, when you're getting percentage, you get, for a pie chart, most of the time you get the total sum, and then you get a particular slice divided by the total sum times 100 mathematically. And then it will give you the percentage or the portion for each. But now in this case, you command R to do it for you directly. That is what this one implies. Last time I said that in case you put around, here in this case, there are two commands embedded. In case you put round, it implies you're commanding R that to give you a rounded off figure, such that you maybe in case you're having, let's say like 35.8. So when you say round, then R will give you a rounded off figure. I went further and I commanded R that, you know what, include also percentages on my figure. So in case I run this on top of what, because we already have this line, I've explained it, this line as well for concatenating. And now when you come to this one, we are just creating percentages, all right? And now, if you've created the percentages, you can even check what are the different percentages. We already have them. And you're like, but I want a plot that has got the percentage on it. So what you do, you use a command paste that you want the percentages to be attached to the labels. So within this command, you start with the labels first, and then the percentages come next within the argument, such that your labels look with the percentages. And then I'll run that line as well. Okay, and it won't give you anything because you want to see everything within the output. If you want to see the percentages on, you create lab. And then yesterday I talked about the separator argument. This line, what we are doing, we are adding a percentage symbol on each label. So it's still with the paste. I just used this, and then I added other arguments, or I can put everything in one line. And then I run this. So let's check. At the end of that day, I use again my pi command. the slices, the labels, call them that. And then I want, I've commanded it to give me a rainbow color, call is color. And then main is the title that you want. So I click on that and then I run. So what happens this time around? In case I zoom, this is what it does. Sometimes maybe you're writing out your work and you want to put things together. So it will bring the percentages attached on the attribute. For example, we've got ideal and the 39%. Okay, I'm going to pause and check if we're on the same page. Hmm? Okay, I'm going to repeat that for the last time such that we are on the same page. Okay, we said initially that in case you don't want to use a bar plot for categorical figures, you can use a pie chart, and we still use a command pie, okay, in R. But you can do a lot of things within this particular command. For example, you can add the attributes, you can put the numbers, you can put the percentages, right? And that is that we are doing. So what I did, remember where I picked these figures from my table, the table that it's quality. I'm considering quality. Remember I had table, and then from my table, I wanted to get the frequencies. Diamond, from my diamond data set, give me quality, okay? So that is all these figures. It is where I picked them from there. It's not that I got them from somewhere else, because I specifically wanted a pie chart for diamond. I went ahead to add the labels as I described, that for 39, it is this one attribute, that for 35, it is premium. The next step, I said, okay, I want to create percentages for each. You know, when you're getting a percentage for a pie chart, in our case, if you're considering 100%, then it implies that you're supposed to first get the sum. Okay, this is the sum for the slices: 39 plus 35 plus 13 plus 8 plus 5. Okay, then I get a portion for each, divide by the total sum. So this line, it's the one that's giving me the percentages, and then the round off, like I explained, for it, it just rounds off figures. Now, when I move to the next line, I use the paste command. Paste, in this particular case, I'm like, get my labels, the labels that I mentioned earlier, they are here, and then on them, add the percentages. On my labels, the ideal, premium, very good, good, and fair, add the percentages, because the percentages are here. I've already calculated that. But on top of that, go ahead and add for me. I can bring this line again, and I put it in the upper line in case I want, and such that I've got this the same thing. So then I like, but still after getting the labels, putting the percentages, also put indicate for me the percentage sign, which is the one that I'm having here. I'm putting a separator. Remember where we said yesterday that SEP is a separator. In this particular case, it's just separate them in tabular form. And what I did, I ran this line. Okay, the different labels that I'm interested in. I wanted to create the percentages, which the object that I created is PCT. If you see that you don't want these objects I mentioned, you can change them. Okay, add on the labels, add percentages, all right? And then also add for me a percentage sign. And then finally, draw for me the pie chart, which we are having here. Okay, I'm going to ask again in the chat whether it is now clear. I'm going to stop. Okay, now let's proceed. There are different options on how you can be able to draw a pie chart. You can also have a 3D pie chart, but I'm leaving that for you. You can try out that command, but all you need to know that for you to have a 3D pie chart, you need a plot trick package loaded. If I'm hoping that you do that, but it's the same thing, and whatever that we have, what I did before, it's the same. What is changing, it's only this part here, the explode, which is equal to 0.1. For it just shows you the size of the pie chart that you're going to have, but and it gives you in form of a 3-dimension. But it's the same thing, only that now instead of using pi only, we use a pi 3D, and then you see how the labels will look like. Just to take you through, but I've already explained the other lines. Yes, in case I zoom out, this is how the 3D pie chart will look like. But you can take some time to it. And then lastly, can we continue? Have you gotten the concept of the pie chart? Okay. Okay, great. I'm winding up. Sometimes this is the part I'm going to do for the last one, and then I'll stop such that my colleague sets in. This is for the case in case you want to do, you want to plot two variables. For example, in case you're having a numerical and then a categorical, you can use the command box plot, and then you type what you're having here is the X and Y. My Y, I considered price, and then that price, it's a function of quality. This is the smile sign that I'm seeing that I've put. Because when you check in the help command, for example, if you ask help, and then you type box plot, you run. When you come to the help command, it will show you the usage that you can start with a formula, and then you start with a formula. Now in that particular case, my formula is at price, it's a function of quality, and then you put the data. My data is diamond, then you can put main and so on. Whether you want to label it or you don't want, you can put an X label, a Y label, and so on. And then you can also run that, and it will give you, you check how it looks like. And that will be how our box plot will look like. And still you'll be able to tell. Remember what a box plot does. It gives you a summary statistics, the minimum, the maximum, the median, and then, and here you can see the different outliers. But more details will be given as we move on. And here you can be able to see that among my different attributes, which one had the highest price. And depending on the look of the figure, you can say that good had the highest price amongst all the other attributes. Okay. Have you been able to produce the box plot for price and quality? Can you try for the box plot when you're plotting price as a function of quality? So you put price then a function sign quality, indicate the data, and then you can put also your title. Okay. Okay, for now, I'm going to stop here. And then in case there are some things that are not clear, I can clear them during the office hours. I'm going to now let my colleagues, Professor Susan, to take over. And this ends the case of my part for module 1. Thank you. Professor Susan, the floor is yours. I've removed.
Because you don't have to show. Let me see.
You can ask if they hear you. You can ask if they can hear you.
Good morning, participants. I hope you had a nice time with Dr. Helen with our exploratory data analysis. I have seen that you've drawn nice charts and you know how to get percentages. Now we are going for a cool down moment. We are going to do some theory. Then after the theory, we will go back to R. Now let me know if my voice is clear. If it's not clear, then I will enhance the voice with the speaker.
It's echoing.
You need to leave the microphone. Okay. That there is an echo.
Yes.
The speaker.
You can ask again.
Is the voice clear? That there is an echo. So you leave the.
No, we tell us it's good.
Is it okay now?
If it's okay now. Please go ahead, it's okay.
Yeah, you need to stop sharing. I am looking for my document. Did I close it? Oh, I had closed it. Okay. Okay. We are going to look at data management, basically using R. Microsoft will be just a hint, but yesterday I sent out materials for today, and we've also sent out a link in the chat, and even the question and answer, we've also sent out a link. So please download the materials because there is an exercise that we are going to do within this lesson or during the lecture. Okay, designing a spreadsheet. We know that we are all interested in having a very good spreadsheet. We are all going to collect data, or we already have data. But before we go to collect data, normally the practice is that you must know how to analyze your data. So when you write your concept and then you develop your full proposal, you should know how to analyze your data And later on, when you're analyzing your data, you just modify what you've already suggested. So once you know how to analyze data, then you will know whether the spreadsheet you're going to design is for a survey or an experiment. So prior to data entry, we construct a spreadsheet in MS Excel or Microsoft Excel. Prior to data collection, we also organize our data files for storing our data and our write-up. So if I'm doing my PhD, I'll have my main folder called PhD. Then I have subfolders. One would be called concept. And then my files will be named according to the dates when I made some changes, so that I do not forget what I did yesterday and today. Then I'll have a folder called proposal. That is where my proposal will be stored. I'll have another folder called data collection. That is where my data sheet will be stored. And also that is where we will have our data collected stored. Okay, then after that we have a folder for data analysis, then write-up. Then you also, as a practice for all MS students, this is outside the training, you need a notebook. This notebook, wherever you meet your advisor, some people are called advisors, supervisors, major professor, whoever is supervising you, make sure you write down whatever they say and store it in that notebook. When you go to the field, make sure you note all the mistakes you've done, so that when you come back to enter your data, those notes you've made in your notebook are going to be so helpful. When it comes to discussing your results, the errors you made in the field are going to be very helpful. And in that notebook, you also draw your time frame. The time frame helps you to achieve the objectives of the PhD or the master's. So everybody should have a time frame, irrespective of whether the supervisor has told you or not. It's very important that in August, I would have finished my concept. In September, I would have started data collection, etc. That is just notes for you to use when you are writing up. When it comes to organizing and designing computer files, make sure you store them in several places. You can store them on your laptop, on your hard drive, you can store them in the cloud. You can send an email to yourself every time, so that your files are securely stored. Because somehow, I don't know what happens to most of the PhD students, their files get corrupted. Their files disappear. The computer gets a virus. So you shouldn't be one of those. You should be smart and store your data in different areas, okay? So for the experimental, we are going to look at the experimental data sheet. There are three main components. We have experimental details, design factors, and measurement variables. Under experimental details, we have things like project name. For example, the project is SDM, Scientific Data Management, and its description. It involves use of R for data analysis. The experimental name could be complete randomized design. We are assuming that all the background information can be controlled. And then the leading scientists, if you do not have anybody, then it should be you. If you have collaborators, probably they can be your supervisors. If you're under somebody's study, then the leading scientist is the person who wrote the proposal. The collaborators are the funders, the people who are funding, and the people also on your PhD committee. Location is where you're going to conduct your study. Design is the type of design. We shall look at type of design in the next session, but the designs we have: complete randomized design, randomized block design, incomplete block design, Latin square. There are so many depending on your objective. The data collector could be you or another person, but I encourage you to be in the field, because from experience, when you get out of the field, even the rats climb the trees. I've heard that saying. They say, if you get out of the house, even the rats climb on top of the roof, meaning that they will do anything they want. So be in the field and be there to collect your data. Then when mistakes are made, you're answerable and you know what to do. Entry technician, it should be you mainly, unless if you have lots and lots of data. Data checker, once you enter your data, you need to clean it. To be able to clean your data, we shall see that, but the data checker could be you, or if you're under somebody's project, maybe they've employed somebody to check the data, so you fill in that information. The design factors, sorry about that. The design factors include columns that identify the block number, plot number, and treatments. Every column must be uniquely identified. For example, in a randomized block design, which I have abbreviated as RBD, we have two columns that are specifically needed: the block and the plot. So if we have 4 blocks, then we are going to have the block as the variable name, and then we'll have 1, 2, 3 as our block numbers. Then we also have plots within the block. So the block is the bigger thing, and the plot, those are the small sections where we apply our treatments, as we shall see when we talk about designs, different types of designs. Now, the third part of the spreadsheet is the measured or measurement variables. What you're going to measure. For example, if I want to find out whether you people are understanding R, my measurement would be how many people are able to draw the box plot, how many people are able to draw the pie chart. That is the measurement variable. The measurement data or variable could be qualitative, it could be quantitative. Okay, so each column has a title section at the top describing the contents of the columns and the details, including the units of measurement. That's very important to include in your spreadsheet, your original spreadsheet. Just like I saw some comments, how can I get my original data? So your original spreadsheet is kept securely once you've cleaned it, and it has all that information, units of measurement. Make sure you put the standard units of measurement. So if you used grams in the field, you will come and include grams in your spreadsheet, but make sure you convert it to kilograms, because the standard unit of weight is kilograms. So you do that within your spreadsheet. And make sure that the names you give to your columns are a little short, because most statistical software require names that are of 8 characters or less. Am I being very fast? Can I see somebody saying no or yes so that I can regulate my speed? Is it okay? Is it understandable? Okay. Okay, some people are saying it is fast. I'm going to reduce a little bit. Others have said it's okay. Okay, very good. Let's continue. So the worksheet should have one row for each unit. That means the plot, subplot, etc. I'm going to show you how a worksheet looks like. So here I have a worksheet. I sent all this to the materials. So I'm going to call it from the Word document section. So give me 1 minute. So here is a spreadsheet. Is it clear from your side? Okay, excellent. So this is a spreadsheet that I downloaded from the internet. So what can I do? Is it okay? Should I go on? Can you see the spreadsheet? They are saying not, still loading. Okay. If it's still loading, then I'll give you. If not, I can go and share the Word document. Let me see. Let me just go and share. Let me go and I'm going to pick one from. Oh no. Okay. Package, slides, experimental data sheet. Has it opened? Not yet. Can you imagine saying a read me only? Okay. Now let me go back, share. Okay. Is that okay now? Yeah? Yes. Yeah. Can you see the screen that has been shared? I'm able to share the Word document. Is it visible? Can I see some comments? Okay, thank you. Excellent. So this is the spreadsheet. I had just hyperlinked it into the presentation. So you can see that I've been talking about the experimental details here circled. You can see the project name, the experiment, the principal scientist, collaborators, location, it is Western Kenya in Maseno. The design is randomized complete block design. We will talk about this. We have an objective for the study, the date of collection. And down here, we have the treatment factors, which are called design factors. We have the experimental plots, the blocks. If I can go up a little bit. So if as I move up, you can see that we see the plots, the experimental blocks, type of mulch, mulch intensity, sample dry weight in kilograms. So here, down here, you can see a plot, 1, 2, 3, 4, 5. In block 1, there are 5 plots. Then type of mulch, you can see different types, abbreviated. Intensity means how much has been applied for each. So this shows you the design part. And then we have the measured variables. They are measuring the sample dry cob weight. Cob is the corn or maize for some people. And then they have sample dry weight. Then they have the total air they receive after drying, the weight of maize. So these are the measured variables. So this is how you design your spreadsheet for experiments. Now, for surveys, I'm just going to give a hint because apparently I couldn't see my survey spreadsheet, which is well organized. We do not have treatment factors in a survey, but we have things like ID number, household number, respondent's name, interviewee. So those are the things which go on the left of your spreadsheet. And on the right of your spreadsheet, the measured variables are things like gender of the respondents. We have how much land they have, their income. All those variables you're interested in will go into the measured section. So if there is no question, I am going to go to the PowerPoint. Okay. So let me go to the PowerPoint. Hopefully you've all learned how to design a spreadsheet. And we are also going to briefly look at the survey one and then go to another section. Okay. So we share again. Huh? Have I shared? Yes, okay. Why do I have edit mode? It's good. Mouse, I've gotten it. Okay, so we've seen the design factors. We've seen an example of the spreadsheet, how it appears. And we are going to go to another section. So I'm going to briefly talk about the survey spreadsheet. Okay. So in the survey spreadsheet, we do not have design factors like in experiments, but variables can include site details. So for example, if my site is in Benin, then I'll put in Benin as my site detail. Then I'll give the exact location, that is the village, interview ID, which will include probably the number of the interview or the name of the numerator, that is the person collecting the data, date and time of the interview. Then we can also have the name of the person responding to the questions. So these descriptors cannot be entered prior to data entry. So we enter these descriptors and then we wait for the details of the other part of the data to be able to capture our spreadsheet. Okay, so let's look at the data structure. So a structured survey with numbered questions has columns for each question and a row for each respondent. So when you have a spreadsheet, you will find that the rows represent each household and the columns represent all the variables in your questionnaire. So this makes it easy to analyze your data. So, unlike in an experimental setting where we already have our predefined treatments and blocks, etc., in the survey, we need to do what they call coding. I'm going to talk about it because I thought probably there are some people who are going to do surveys and may need this information. So basic coding implies that we have materials which can be put into nominal level categories. And my colleague Helen talked about nominal. This should reflect the objectives of the activity. It is not necessary to code information that is not relevant to your objectives. You need to prepare a code book. I believe everybody is saying, what is a code book? It's just like any other book. You get a notebook and then rewrite your questionnaire. But as you rewrite your questionnaire, your questions, you're going to code those questions. So how are we going to do this? Why do we code? Before I show you how to write the questionnaire, we code because we want to transform our nominal and ordinal categorical data into numerical information that can easily be analyzed by the software. But those codes do not have any meaning. For example, if we have our question as gender of respondents and the responses are male and female. So for ease of entering in the software, we give the codes male we give 1 and female we give 0. It does not matter, we can interchange and say female 1 and male 0 because those numbers do not have a meaning. Don't say that 0 means there is no male or 0 means there is no female. No, they do not have a meaning. We just use them for ease of analysis. So in your code book, all questions that have categories, for example, gender, religion, location. For example, you might have 3 districts where you are moving to within Benin. So you code those districts. All that information, you give them codes for ease of remembering when you're analyzing and for ease of analysis. Okay, so do we understand what a code book is? Or we need to... Do we understand what a code book is? Excellent, that is really good. Somebody is saying talk about again. Okay, I will just briefly talk about the code book. So I say that you have your questionnaire set out. And remember, the questionnaire is developed following your objectives of the survey. So you have section A, which has, people call it social demographic information, others call it demographic information. Then section B is objective 1, section C is objective 2, section D is objective 3, depending on the number of objectives you have. So then you develop a code book. This can actually be developed before you collect data. So you get a notebook and rewrite those questions in your questionnaire, rewrite them in the book. But as you go question by question, you give them numbers to the categories of responses. For example, if one of the question is location, that is location of the respondent, then you're going to have 5 locations and you're going to code the first location as 1, the second location as 2, 3, up to 5. If the next question is gender of the respondent, then we are going to have female and male. And then we are going to code female with 1 and male with 0. Okay. And then after that, we also mentioned that the numbers you attach to your categorical information do not have any meaning. So you will not go and get average female codes. No, those ones do not have any meaning. They are just numerical values attached to categorical data for ease of analysis and entering in the software. So the code book is a guide for you as you enter your data. You'll be able to see, oh, location 1 is coded 1, location 2 is etc. Then for those using software like SPSS, can actually transform the code book into SPSS by defining the variables and indicating those codes there. But if you already don't have a code, that means you're going to first of all code the questionnaires, then again enter them into your software. Okay? So this is just for ease of analysis and entry into your software. Somebody says I should share a code book. A code book is just like any other book you have. I really don't have a code book here, but probably I can turn to the chart, the whiteboard there, and try to write for you on the whiteboard. So I'm going to move to the whiteboard, the manual whiteboard, not the one in the, okay? So one thing I'm going to ask you guys, do you see my whiteboard? If you can see the whiteboard, then I'll continue. If you don't see the whiteboard, I'm going to improvise. So can I have responses of those seeing the whiteboard? Okay, so assuming this is my code book, and then I am on the left and I'm right-handed, so excuse me if I write very badly. We have the first question, so we are going to call it variable 1. It's ID.
Prof.
And here ID is going to be number.
Prof, I'm going to stop your screen from sharing such that the participants are able to see your whiteboard.
You see the handwriting, or I make it bigger? Let me go down. V1 is ID, and this one is number of respondent. Okay, so let me assume that the next one, V2, is gender. So my name is going to be gender. And the codes are going to be 1 for female. I'm going to start with female because I'm female. And 0 for male. So that's how your code book is going to be. Is that clear? Can we go on? Or you need more explanation? So we can have variable 3, but I don't think you see the lower bit of it. So let me go to another page. So we are going to have variable number 3, and it's going to be location. And under location, I'll have 1, 2, and 3. So I'll say Uganda, because I'm in Uganda. Then I'll say Kenya, because it's East Africa. I'll say Benin, because it's Africa. Okay? Then 4, Swaziland. Okay? So this is how you code your data for ease of data entry and analysis. Is that okay? Can we now continue, or you have a question? Okay, thank you very much. So I'm going to go back, and then we sit and do something else. Okay, so let's continue. Okay, one thing I also wanted to talk about later. Okay, so there is something that most of you experience in survey data collection is what we call multiple response. For the rest of the questions we've seen, like location, somebody will be in one location. They can't be in two locations at the same time. But there are some questions that you ask. For example, when we sent out our questionnaire, you said people had so many expectations. Somebody had 5, another one had 4, another one had 3. So we call those multiple response. So if you do not capture the multiple response well during the data collection section, during the data entry part, you're not going to be able to analyze multiple response. So here I've given an example that multiple response involves questions whose respondents can choose a number of relevant responses. For example, if you are using an improved fallow on your land, please tick from the list below any reasons that apply. So one person will say, do not have any land of my own. And then do not have any suitable crop for an improved fallow. So the same person has said he doesn't have land of their own and doesn't have a crop that can be grown in an improved fallow. Then he also said, I don't have money, I cannot afford to buy the seed or plants. He also says, I also don't have time and labor. Okay? So if you have, that is one person, then another one says, I do not have land. Another one says, cannot afford to buy seed and do not have time. Another one says, do not have suitable crop, cannot afford seed, do not have time. Okay? So the way you enter variables that have categorical information well coded is different from the way we enter multiple response. So there are 2 ways, there could be 3. I'm going to explain 2 ways. Okay, we have the simplest one. So in the simplest format, you make sure all the responses you've gotten. So you look through all your questionnaires after data collection. Note down all the responses. If there are 10, then you're going to create 10 variables. So one of the variables will be no land, no crop, no money, no time. So those are the different variables. Now, if somebody says no land, the response is going to be yes. If the person says doesn't choose no land, the response is going to be no. So when you're entering in the spreadsheet, you enter 1 for yes and no for somebody who has said no. Does it make sense? Okay. Okay. Okay, so I'm going to repeat. The type of entering data I'm explaining here is called multiple dichotomy, where you have yes and no representing the responses that were indicated by the respondents. So in this example, we had only 4 responses, and for each response, I'm going to go backwards. So you can see for each response, we are going to have a yes or no as our response. So if I say, I do not have land, my variable will be land. So under land, my collected data, I'm going to enter 1 if that respondent said yes, or I'll enter 0 if that respondent said no. Then I go to the next respondent. But if he gave more than 1 response, I'm going to go to the next column, which says suitable crop. And if that person said no, then I'll put 0. Then the next column will be no money. They cannot afford to buy the seed. So under that, if they ticked yes, I'll put 1 for yes. The next column will be time. If it was ticked, then I also put a yes. So that is only one respondent. Then I go to column number 2. Okay. So this is one variable. I'm just checking the chart to see the questions which are there. And people are saying, how about if you have 10 variables? Yes, if you have 10 variables, that's what you're going to do. Otherwise, you're not going to be able to analyze that data. You're going to do that same thing. You're going to have 10 variables entered, and then you put your yes and no. Now, if you have 10 questions, remember, and they all have 10 responses, you're going to have 10 times 10 columns in here, depending on your questions, okay? So that's what is going to happen. Any other question? Yes, I'm checking the question as okay. In multiple response, each response is taken to be a variable of its own. And then when the person ticks that response, you put yes and no, depending on whether they've ticked or they've not ticked. Okay. Okay. Somebody is saying internet connection is very bad. I think here in Uganda it's drizzling and the internet is unstable, but I believe everywhere, I'm not sure whether it's drizzling, but probably there should be something interrupting the internet. Oh, you want an example? Okay. So what I'm going to do is to shift again to my whiteboard. I'm going to shift the whiteboard and I'll stop sharing. So hopefully you will remember the options. So I'm again at the whiteboard. Okay. Okay. So here I am with my whiteboard again. Now I'm saying that each response becomes a variable of its row. So here is our spreadsheet. Those are the columns, these are the rows. Remember, the columns represent the variables and the rows represent the respondents, each household. So this is a spreadsheet, an Excel spreadsheet. So here we are going to call it land. And here we are going to call it crop. Remember, the responses were do not have land, do not have suitable crop. Then the seed, I'm going to call it seed. The last one, time. So those are the multiple response we had for our question. Okay? Now I said that we are going to code, if somebody says they do not have land, we are going to put 1, and the 1 represents yes. Okay? If somebody says they do not have a suitable crop, I also put 1. If they don't mention this, I leave them, I just put 0, 0, meaning no, no. Then if I come here, this is respondent number 1. Number 2 says he doesn't tick this, but ticks this one and this one. And these ones doesn't tick. Then we go to number 3, respondent number 3. Number 3 says they don't have land, and this one doesn't tick, but ticks this. Okay? So that is how your spreadsheet is going to look like. These are the variables. They've become variables, and this is the ID of the respondent. Is it clear now? Okay. So I'm going to stay here. Okay, now I'm going to share again so that I don't have to move. Okay, some of you are ahead of me, so you're asking questions that are ahead of me. So we said that so that is the first way where we use yes and no, and we've coded our yes and no as 0 and 1, where the 0 is no, and then 1 is the yes. So we go to the next section. The next second way, find the maximum number of ticks from anyone, and then have this number of columns entering the code. So what we are going to do here, so in this second part, we are going to make the responses variables, but at the same time, we are going to make, when somebody says no land, no crop, we are going to include all those responses under each variable as our codes. So how is that going to happen? For example, I'm going to create, let me go back, stop sharing and go back to the screen, the whiteboard. Hopefully it's visible. So I'm going to use one example and one variable. So we are going to say the first variable is do not have land. Do not have land. And under this is the variable name. Under this we have, again, no land as a possible option. Number 2, we will have no crop to grow in furrow. Number 3 is going to be money. They don't have money to buy the seed. Number 4 is time to do the tilling. So this is the second option. The first option, that each of these becomes a variable and you have a yes for those who ticked it and not. In this second one, you make the responses, all the responses variables, but again, under each you list those responses as options that can be chosen. So after that, let me show you how you enter that data. So for variable 1, so this is land as our variable. And maybe we have crop as the second one. So if the person says no land, land was coded 1. But if you also said crop, you also put 2. And if it didn't say time, we leave it as 0. And also this one, if it is, this is money, we leave it as 0 because it didn't mention anything. So the first respondent said he didn't have land, didn't have crop. The second respondent said he doesn't have crop, and then time and money. You see? The third respondent have crop, they don't have all this, they don't have all that. So you see the difference. Here, in the first case, we had a 1 and 0. Here you see all of them appearing, depending on the responses of the respondent. Any question? Can I have questions in the chat? I can read them very fast. Okay, somebody is saying, please repeat that part. Okay, so I'm going to go back. So you remember we had four responses for our question of whether they use the fallow land. That was do not have land, do not have crop, do not have money, and then time. Okay? So in the second way of entering multiple response, we make sure all the responses become variables, and the ticked information, those particular responses again become codes for each variable. So for example, if our variable is do not have land, then all those responses are coded down here for you to choose from. So you have no land as 1, no crop as 2, money is 3, and time is 4. Now, when you come to entering the data, each column is a response. Remember, each column is a response which has become a variable. And if the first respondent said yes, they do not have land, you put 1, because 1 was the code of do not have land. And if you also said it doesn't have the crop to grow in furrow, then you also indicate 2. If you didn't tick the rest, you put in zeros. Then respondent number 2 said they do not have crop and time and also money. So you enter the codes for crop, 2, time, and 4. Okay? So the columns become the variables, and the variables are the responses. I'm responding to the questions. Okay? So if it's clear, let me see some yes coming up. If it's not, then I'll see no. Okay? Okay, so I am going to go back to a comfort zone. Okay, let me share. Okay, there was a question that how do you analyze such data? It's easier to do it in SPSS. That's what I know. In R, I haven't tried analyzing multiple response. That is a fact. But in SPSS, very easy, and things are straightforward. Just like I said, R is so huge. It's a software which it doesn't have things put together in one package. So when you need like to analyze multiple response, you put in Google multiple response analysis. Then they will give you the code. But we shall see what we can do. Okay. But both methods, the simplest one and the second one, give the same result. When you start analyzing, they give the same result. So don't worry that you use method 1 or method 2. Some people prefer using method 2, others like method 1. I like method 1. Okay. And remember that when you put your data in SPSS, you can actually export it to R and analyze the data there. Or you can analyze it in SPSS and then export it to R. Remember, as a student or as a researcher, you don't stick to one software. And at the beginning, we mentioned that the reason why we have introduced R is the situation has forced us to think further, think out of the box, like we've been using GenStat, SPSS, etc. We should get out of that because those are licensed software, and if they are licensed, you cannot teach them online. So that's why we move out of that and go to R. Okay? Okay, let's go ahead. Now, I sent a spreadsheet. I sent an exercise for you to enter only question 1 in the spreadsheet. Why do I do this? I want to find out whether I really need to tell you how to enter data in Excel. So, is there anybody who has entered this data in Excel? You can raise up your hand, and my colleague will give you help you to share. Anybody who did the work, who entered exercise 1? Exercise 1, let me share the exercise so that you can see. So I'm going to stop this. Somebody has raised up their hand. So my colleague in charge of IT, can you let that person share? People are saying they didn't get the material. We've sent the material. The link has been sent into the chat and also email. We've done it twice on email, and we've also sent the chat in the Q&A and also in the chat. Okay? So you should be able to get the material, really. Also, when you are learning, also check the chats, because we keep sending whenever somebody asks for material, we keep posting the chat immediately. So you also keep checking the chat, okay? And we also sent on email. Last night I did that, and I also sent to my colleague, and she did it this morning. So we've really tried to make sure that we are the same page. But for those who haven't received, we are very, very sorry. Okay, let me share the question so that probably I can give you 5 minutes and you will be able to tell me. Okay. Okay, so I have shared the data that, I mean the spreadsheet or data with the data that I sent. This is the spreadsheet. So I do not want us to move from this stage before we know that you all are comfortable. If you go to collect data in the field, will you be able to enter that data in Excel? So only for task 1, I'm giving you 5 minutes. Can we try to open your Excel wherever you are and enter this data? You can enter the first 6 columns with their suitable variable names. Okay? So if you look at this, we have plant that goes up from 1 up to 13. And then we have bottom. Of plant 1, we collected bottom and top. Plant 2, bottom and top. So put this in a spreadsheet, and we'll have 1 or 2 volunteers to show us how they've entered their data. So 5 minutes, you have 4 minutes. Let me mute. You have 2 minutes left. So whoever is ready can write in the chat, I'm volunteering or I'm ready. What are you supposed to do? Enter the data in an Excel worksheet, Microsoft Excel. Godfrey Arena is ready. Okay. So Godfrey, be patient. I want the 5 minutes to end, then you're going to help us share. Oh, there are so many. Josephine says is ready. Chibuka Muhammad is ready. Alice Nachiemba. Mariam is ready. Godije, I'm sorry if I pronounce your name wrongly, is ready. Yes, I can see so many ready people. I'm requesting the IT to allow Godfrey Arena IT to share. So I'm going to stop sharing if that is okay. You've all seen what task number 1 is. So I'm going to stop sharing. So Godfrey was the first one to share what he has done. Then we also pick one lady. I saw Maria. Maria, you can also share. So let me inform the IT person to allow. IT person, please allow Godfrey Arena Itwe to share what he has done. Yes. David Amitu, can you allow Arena Itwe to share, please? Oh, Waswa. Please, somebody doing IT who is controlling our session this afternoon, can you please allow Alina Itwe to share? Godfrey? So participants, I am very grateful. I saw so many people saying that you're ready, which is very, very good. And I've been checking the Q&A. So people are saying I'm done, I'm ready to share. There are very many people ready to share. That is really encouraging. And I wish we could also share at the same time, but it's not possible because of technology. Let's have the first person share with us. Then we'll have Maria share so that we balance gender. Okay. I'm saying that allow Godfrey Arena Itwe to share the spreadsheet. David, allow David,
Yes,
you
please.
hear me? Allow
Yes.
Arena Itwe Godfrey to share the screen. He's going to share what
Okay.
he has to show that he knows how to capture data in Excel worksheet. And then after Godfrey, we'll have Maria.
Renata Godfrey is not in. Okay, I've promoted him.
Yes, Helen, I have seen that comment. You're saying very good interaction. Thank you. Somebody says it was called unaware. It happens. Okay. I am looking at your comments. And very many people say they are done, they are done. They cannot use the mic. We only have to use the mic when the person allows us. So let's see what Godfrey has for us. Okay, Godfrey, you can, yes, make those values a bit bigger. You can either use Zoom or make, yes, that is very good, Godfrey. Okay. So I am, Godfrey, I'm going to ask fellow participants, is that the correct way of entering the data? He has plant, then bottom and top. Somebody says yes, 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 yes. Okay. So all of you entered your data like that? Am I going to assume that? Somebody says, I don't think so. Another one says, no, 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 no. Sure? No. Okay. Yes. Okay. Godfrey, it is okay. You don't need to move the cursor. Says the variables are too long. It depends on the analysis to be performed. Then somebody says no. Okay. That was a good comment. Depends on the analysis to be performed. Okay. That is also another question for you people. I'm going to ask you, what type of analysis can we apply to this data? So let me have some answers. What type of analysis can we apply to the data the way Godfrey has organized it? What type of analysis, what statistical analysis can we apply? Somebody is saying average, okay, that is okay. ANOVA, let me put it down. ANOVA t-test, good. Mean, yes. Regression, no. T-test, yes. ANOVA has a star. ANOVA using CRD has a star. Okay, thank you for the responses. Now, using Godfrey's way of entering data, and depending on the software you're using. Okay, now let's pay attention here, ladies and gentlemen. Those in the chat, let's pay attention so that you don't miss this point. I'm saying that now the way Godfrey has entered the data depends on the software you are in. Somebody said paired t-test, excellent. Okay, I have asked a question, what type of analysis can we apply? Now, the type of analysis depends on the objective. The objective is to find out the difference in number of seeds or weight of seeds in the bottom and the top. So I want to find out whether those two are different. And we are collecting bottom and top from the same plant. We are collecting from the same plant, meaning that the two values are correlated. One depends on the other. The same plant is giving us seeds at the bottom and top. So there's dependency. And for us to be able to analyze that data, one person said paired t-test, and that is the correct analysis, and I'm happy about that. Somebody who said t-test in an exam, I'll give you half the mark because you have an idea but not specific. So in statistics, we are supposed to be specific, and the correct analysis would be paired t-test. Now, somebody will say, why not ANOVA? Analysis of variance can only be used if we had more than two groups. Now we have only two groups. If we have three groups, maybe middle part, then we would have applied analysis of variance. Somebody says descriptive, very good. The first thing you do before you analyze your data is descriptive statistics, so that you have a feel of what is in your data. You know the trends, you can draw some graphs, you can do the means to see whether the mean for bottom is different from the mean for top. So if you see they are different, then we now want to use the paired t-test to find out whether those means are statistically significant. Okay? How about regression? No. Regression is used to find out a relationship between two variables. For example, here we have bottom and top. Depends on the bottom, or the bottom depends on top. which is going to be our independent variable, we must have measured, and the dependent variable is the one which varies a lot, which is affected by so many environmental and experimental variation. So if we wait, oh, somebody is saying that a little confused. We shall sort that out. Okay, so we are going to finish this part. We are saying that regression, when we want to do a simple linear regression, we want to have our dependent variable well specified, depending on the objective. You remember, our objective is to find out whether there are differences in the weight of seeds at the bottom and top. So if you want to find out differences, then regression doesn't really apply. Now, if we wanted to find out whether there's a relationship between the weight of the bottom and the top, then we would think about a regression. But in this case, our dependent variable would not be weight again. So in this case, we are going to reorganize our data and make a third variable called plant part, so that we have bottom top becoming the plant part. So plant part now becomes our independent variable, and we are saying that the weight of the seed depends on the plant part. You can see the difference now in analysis. So the way he has organized your data, we can easily analyze it with a paired t-test. And also it depends on the software you are in. Different software want your data organized differently. Okay, so if we wanted to use regression, our objective should be related to regression. We want to find out a relationship between the plant part and the weight of the seed. So now plant part becomes our independent variable, and then weight of seed becomes our response or dependent variable. Are we together about the type of analysis and how you choose and how you organize your data? So depending on the software where you are, you have to change the way you organize your data. Okay. Maria, do you think we should have you to present, or you had the same thing? If you had the same thing, then it's okay, we can go ahead. You can put a yes in the chat, Maria. I had the same thing. Okay, good. Now me, I have something different. Okay, David, you can stop Godfrey from sharing. Thank you, Godfrey, for trying. That was a very good trial. Thank you very much. And you can now remove your video. I'm going to share a spreadsheet entered differently from Godfrey. Okay. Give me 2 minutes to do that. Okay. Okay, so I hope you can see this. I have weight as a variable, and then I have plant part as a variable. Then I have plant number as a variable. Remember when I said when you're entering data, your factors should be on the left and the measured variable on the right. So you can see on the left, I have plant number and plant part. And then here on my right, I have weight, which is the measured variable. So this is another format you can use to enter your data. And now with this format, you can analyze, you can now get a relationship between weight and plant part. You can also use the paired t-test as well. So that's what I wanted to show you. So you can now, in your free time, go and practice. You have the Word document with different data sets. Try to enter them in Excel. Tomorrow in the morning, we can see like another 2 people showing us task number 2, how they try to enter their data. It's very good to know how to enter data. Then your life will become easier when you're analyzing. Okay, so please, if you do not have a question, say I should continue with the PowerPoint. So can I see yeses or no so that we can continue? Yes, we are going to send the link of material for today and you'll get it. Okay, let me continue to the PowerPoint. I'm going to stop sharing the spreadsheet and I'll share the PowerPoint. Okay. So I must say that I'm very, very grateful for your participation. That was really excellent on your part. You did a very good job and I'm happy. Okay, that makes me happy that you're going, after this session of 6 days, you're going to come out well, trained data manipulators or analysts, whatever you want to call yourself. Okay, let's go on. Data entry and checking. Okay. Now, this part I brought in just... So you must be saying, now where is the R? R is coming. Okay. So data entry. As soon as you collect your data, don't keep your data. Don't keep it for a week. Don't keep it for 3 days. Because you're going to forget some of those things, the mistakes you made. They are very good. For example, when I was doing my PhD, I was harvesting agroforestry tree species. I was trying to develop allometric equations for some species used in agroforestry. So I got permission and they let me cut down some so that I can get bits. But then you have to collect those bits, the bottom, top, middle part, then cut off bits, weigh them. So if you forget anything, you will remember that actually I never weighed this part of that particular tree. So enter your data as soon as you collect the data from the field. Don't delegate your PhD to another person, to a technician, or some people call them data analysts. I'm going to take my data to the data analyst. If you bring me your data, I'll do what I think is right, not what you think on your work. Even when you to the defense, you should know what type of analysis you used. Don't refer to some technician in some lab who did the SPSS for you, the R, when you're stuck with the code. You can be stuck with the code, it's allowable, but always own your mistakes, own your work, okay? Okay, so enter your data. So you can be 2 people entering the data, we call it double entry, and then afterwards cross-check. You can merge those 2 data sets using an ID, and if they match correctly, then that data is correct, or you can run cross-tabulations. Okay? Cross-tabulation is when you draw, I had Dr. Helen showing you how to get counts of quality. Now, if we cross-tabulate quality and color, we are able to know that actually in a particular cell, instead of entering blue, you entered BLE. You see, that is a different color. BLE is a different color. So, but it's a variable, it is a data value you've entered in a particular variable. So how can we capture those? By doing cross-tabulations of categorical data. And once you find there's a mistake, Then you can go back to your questionnaires. One thing I forgot to tell you is that when you're also making your code book, you should have questionnaire number as your first variable. Reason, it helps you to go back and sort your problem out. So you have your questionnaire number up to 100 or 1000. Those are the number of questionnaires you have. So if you've made a mistake on questionnaire number 2, you'll be able to locate number 2. Remember, each row represents the questionnaire numbers and also the respondent numbers. But respondent number is not going to easily guide you back to the questionnaire, but questionnaire number will easily guide you back to your questionnaires. You dig out that questionnaire and correct that mistake. So it's very important to have the questionnaire number as part of your data coding. Okay? So we've talked about cross tabulations, scatter plots. You can draw scatter plots of continuous data. Then you can see outliers. I believe you've heard of outliers. Those are values which are different from the rest of the data. So pay attention to the outliers. They may be correct outliers in that they really exist, or they may be by mistake. For example, instead of putting weight of 20, you put 200. So it becomes an outlier. So check in your field sheets whether the weight of that particular maize or corn was 200 kilograms or 20 kilograms. Pivotal table in Excel, you can construct pivotal table. Remember, we are now entering data. We haven't gone to R. We are still entering data in Excel. You could enter your data in R, but it is a bit tedious. So you need to know your R properly to enter your data in R. You can do it for small data sets, not for large data sets. So in Excel, you can do pivotal table. Pivotal table, I believe all of you know some bit of Excel. And if you don't know, we have Mr. Google, or is Google she or a he? One of the two. Google will tell you what pivotal table is. But it is a table that allows you either to do means, you can get mean of the response, or you can do cross tabulations, or you can get the standard deviation. In the pivotal table, there's a lot of analysis you can do. So that is checking, you're checking to see errors. Do not rush to do your analysis before you clean your data. Because if you bring me your data set, the first thing I do is to do some cross tabulations. If I find a mistake, I just tell you, please take back your data and do what? Correct it. Even when you're just consulting, the first thing I do is to run some cross tabulations. In any software, as I said, don't stick to one software. R can be good in one software, but SPSS can be also good, or GeneStat. So be flexible to learn. Your students, you should be flexible to learn and be ready to teach yourself some things. Don't wait to be taught. Okay? So once you do some cross tabulations, you can highlight out mistakes. For example, you have gender, male, female. Male is coded 1, female is 0. In your data set, you enter 100 for male. When you cross tabulate with the religion, you find there is another group called 100. So you're able now to check where 100 comes from. It's just a data entry mistake. So once you have a clean data set, you've entered your data set well, now you can export it to R and do analysis. There's what we call validation. It is in Excel again. You can limit, you can actually specify the way you want your dates to appear, the codes, how many codes you want per variable, so that when you're entering your data, it is used as a check. If you said male is 1 and you enter 10, it's going to give you, it's not even going to allow you to enter 10. So it is a check in Excel for you to help you enter correctly your data. And also enter things like date, things like units, etc. Okay? Can we continue? Oh, okay. I want to check. Okay. Ooh, I missed some question. Somebody says. Huh? I missed a question. Somebody was saying also explain something, but I missed it. Moved so fast. I didn't get that question. Okay. But if I see it again, I'll explain that bit. Okay, let's move to the next slide. Okay, somebody is saying I should explain normality and homogeneity. Normality is basically done in analysis, not data entry. So once we've entered our data, we've cleaned it, we can now export it to R and do our normality test, which shows us whether our data is normally distributed. If it's normally distributed, it should come up with a bell-shaped curve. Otherwise, we call that data skewed. But it does not necessarily mean the data is bad. No, skewed data is good because there are people who are very short and those who are very tall, we don't think they are abnormal. Okay? So that is what we call skewness. People who are so different from others or values which are different, they tend to pull the variable to the small outliers or the big high values. Okay. Then homogeneity. Homogeneity is an assumption under analysis again, which we are going to look at. We assume under analysis of variance that the residuals after analysis are homogeneous, meaning they are not different, they are constant. So that's an assumption. Okay. Okay, so let's go to the next section. Oh, now let's go to R. Before we go to R, we have something here. Now it's time for R. Hopefully we have some more time. Oh, we have only 18 minutes. Okay, so we are in 18 minutes, we only do a simple thing in R and then we will close down. Expectations of what? Sorry about that. Okay. Now, R commands can be used to design experimental field sheets. So here is an example. A researcher wants to investigate 3 times of raising bread. This is food science. I saw some people who do food science, so I said, now let me bring some example from their field. So there are 3 times of raising bread. So when you prepare that dough for bread, there's a specific time you give it to raise and check whether it is ready. So we want to find out which is the best time for our loaves to be ready. We have 35 minutes, 40 minutes, and 45 minutes. And they are replicated 4 times. So if we have 3 treatments and they are replicated 4 times, from your knowledge of experimental design, how many loaves of bread do we have? We can see in the chart. Let me see the chart whether. How many loaves of bread do we have? We have 3 times of raising bread replicated 4 times. Somebody is saying 9. Let's see. They are replicated 4 times. 12, 12, okay? 12. Let me have more responses. We have 3 treatments of bread. Do we have, remember, bread is our experimental unit. Okay? You will ask me what is an experimental unit. That's why, okay? Thank you very much. One person has said 1. And another one has said 2. Okay, 3 times. Okay. So in this case, the number of experimental units, the number of loaves of bread are 3 times. You have replicated 4 times. So that is 3 times 4, meaning we have 12 loaves of bread that we are going to investigate. Okay. So let's move away from that. So the following R code will create the list of field book, which you will go with to collect your data in your lab. Now, this food scientist will go with this field sheet, and wherever he removes the loaf of bread and looks at it, he records down the height of raising, okay? Okay, so we have this code here. Before we go to R, I want to do a bit of explanation. I know you've been doing descriptive, you've been doing manipulation, and probably some of these things you haven't seen. So let's go through this R command. The first one is set a seed. This set a seed allows you to randomize this experiment. And every time you run this command, you're going to get the same thing. If you don't put anything and you put 0, or you don't put set a command, every time you run this command here, these commands, you're going to get a different randomization. Remember that in a complete randomized design, we need to randomize to avoid bias, and also it is an assumption for us to be able to perform our analysis of variance. That's why we need to randomize. But you can choose. Do you want to have different data sets all the time? Different randomization of treatments to the experimental units? No, we want to have one randomization. This number here, you see 706. You know that the random numbers, if you've seen a random number, so many, they can start from 10,000, depending on the random number table you're looking at. So you can put actually 76. It does not matter what number you put here. You will still get your field sheet. So the second part here, we call this an object in R. And this object, we are assigning it, or it is equal to a factor. And that factor is going to be replicated. And we put them together. I'm just following what Helen told you. I'm not changing the English. This is our language. So this object is going to consist of a factor, which is replicated. And we put all these things together using this C. And these are the minutes 5, and each is replicated 4 times, and we close the brackets. Let me go to the next row. The object is called bread. And these values here are going to be randomized, or the bread is going to be randomized using the object X, and there are 12 of them. As you told me, there are 12 loaves. So that's why we have that here. Then Y is the vector. Then raise is going to be our data set. Now we call object raise. We want to put all this information together in raise. We use this command called data frame. And loaf is Y, there are 12 of them, and time is bread, okay? So we've told this part here, function sample, to randomize these to 12 experimental units, to 12 loaves of bread. That is the second bit. Then we write our raise. We write it to a CSV file. Okay, I go back. Somebody said I should explain the seed. The seed is just a command telling R to generate a data set called CRD, starting with this random number. We can put 10, we can put 20. That is if you don't put set seed and you start from X, then every time you run this command, you're going to have a different randomization of these treatments to the loaves of bread. That's why we need seed, so that we have a constant randomization. Now I'm assuming you're going to the lab, and this is your experiment, and you do not want to have different randomization every time you go to the lab. All the data sheet is filled up. Now you want another data sheet. You come, you didn't put seed, you get another randomization. You understand what I'm saying? So that's why we set seed. Okay. Now from seed, we want to specify the treatment, which is a factor, and replicated 4 times. That's why you see each equals 4. and there are 3 treatments. So bread is time. Time is going to be randomized. That's why we have this function sample, and there are 12 of them. We also have y, which is the loaf of bread, and they start from 1 to 12. Now, we are going to put all this information in our object arrays, and we are going to use the data frame function to combine the vectors y and bread. Then we write that information onto a CVS file so that you can now print it in Excel and take it to print it out and take it to Excel, print it out and go to the lab. Okay, so this is the right command which you've already seen. Raise is the object from the previous line. File, we call our file raise CVS. The rows equals false. We use the head function to print out the first 6 rows of the data. And I got this source. Remember always when you're writing these things to quote where you got them. So I got it in Lawson 2015. It is actually a very good reference for those doing experimental design. It has lots of experimental design there, so you can always refer to that book wherever you can. So the next slides are explaining what I've been saying. The R command, the command factor creates a vector of factor levels for rise time and stores it in X. The function sample randomizes the order of the factor levels and stores in bread. Then the data frame function combines the 2 vectors bread and y as columns that are stored in the data frame object arrays with column heading loaf and time. Then this write function is going to write raise as a CVS file, and you can find it in your directorate. Now let's go to R. I remember I sent out this script. So for those who have the script, just open the script and we are going to run the script in R. Oh, I'm told I need only 5 minutes. Okay, 5 minutes, we can do whatever we can. We can open R and okay. With your permission, maybe I don't need to get permission from you. Somebody here is harassing me. Sorry about that, but she's told me that there are questions that you need to answer, not as a must, but as a request. So we are going to stop here, and I encourage you to go to R, open that script and run it. You don't need a data set. Just run the script, check your directorate. If you see rise, then you would have done the correct thing. Check rise whether it has bread and time of rising. So I think we will end here and we will start from here tomorrow. I am so grateful. I have enjoyed myself. I don't know about you guys, but from the answers I've gotten from the chat, I believe you guys have enjoyed yourself, and I believe you've benefited a lot. If you haven't benefited, please send me a comment so that we can improve. Lawson is a book. Yes, Lawson is a book. But you can download it from internet. Just put in experimental design using R and you will get a lot of material. There's also another PDF by Felipe. It also has a lot of information on experimental design. So please, in these remaining minutes, I'm allowing Helen to share something which she needs effective. I didn't see anybody dozing, so thank you so much. And bye for now. Okay, video
and thank you, Prof. I hope that you're enjoying all the sessions that are going on. we just have a simple request before we end this session for today. I'm sharing the screen and we encourage everyone to participate. There are just few questions that we need your attention on. And please type the responses in the chat box, then I will pick them. So we are asking, what have you learned so far in this training? And what can be improved in the next session? Then was today's session helpful to you and why? So we need to have the feedback from you such that we're able to improve in the remaining days and to make our interaction much better. Still a reminder, we are having office hours at 2:30. For those who might be having some issues here and there, we'll be happy to support you such that we all, at the end of the training, we've benefited equally. So kindly get in the chat box and respond to the questions that I've shared with you. Thank you. Okay, let's start with question 1. Let's start with question 1 such that we're able to get a feedback properly. What have you learned so far in this training? Question 1. What have you learned so far in this training? Okay. Thank you so much for the different responses. To make capturing your responses better, when as you're responding, just start with a 1 and then then do the writing. Now we are moving on to, in case of question 2, what can be improved in the next session. So you type 2, then dot, and what you think that can be done to improve these sessions. I want to see the different responses for what can be done to improve the next sessions. You type 2 and then I put what you think that can support the facilitators to see that you get what you expect to get at the end of the day or at the end of the training. And then number 3, was today's session helpful? And if it's the case, why? If no, why? All the answers are correct. The main purpose is to help us to see how we can make the training better than what it is now. I also encourage you to check the different links that we've been sending, such that for those of you who are saying that you haven't yet gotten the material, you equip yourself with them, you're up to date. We've set Google links whereby you can go there and download the material with you, because some people say they miss getting the emails here and there. But in case you go to the links that we've sent, then anyone can access those materials. I think we can end. Sorry? Kindly provide answers for all sessions, question 1, question 2, and then question 3. I think they're done.
There are some questions.
Okay, as you're moving ahead to attempt the questions, we're looking at what you've learned so far in this training, what can be improved in the next session, and was today's session helpful to you? If yes, why? And if no, why? We just want to use the feedback such that we make the training more interesting or more resourceful for you, such that by the end of Saturday, you've got a take-home message about R as the training is ending. Okay, so in case we are done, we are going to pick the responses, and I wish you the best of the afternoon. In Uganda, it's afternoon. Maybe it could be a different timeline in other places. And then we can talk at 2:30. We can take a break and then we talk at 2:30 again.